Hi everyone, this is chapter 13 on viruses, virions, and prions. So let's begin with talking about some of the characteristics of viruses. They are teeny tiny, minuscule. They are acellular, meaning they are not a complete cell. They are infectious agents and they have either DNA or RNA as their genetic material. They cause infections in humans, animals, plants, and even bacteria. They cause most of the diseases that plague the industrialized world. They cannot carry out any metabolic pathway, nor can they grow or respond to the environment. They cannot reproduce independently. So instead of being able to do all of those things, they actually are going to hijack one of the host's healthy cells and use its metabolic pathway to increase their numbers. They have no cytoplasmic membrane, they have no cytosol, they have no organelles. They are basically a container for genetic material. They do have extracellular and intracellular states. So viruses are unique in that they're not really alive. They're in that quasi no man's land of not living because they can't do a lot of the things I just mentioned. They can't carry out metabolic pathways or grow or reproduce. They have to take over a healthy cell to do that, but there's not really any other place in biology to put them. And they do cause a lot of diseases, so we definitely want to talk about them in microbiology. The extracellular state of a virus is called a virion. It has a protein cap or coat called a capsid that surrounds a nucleic acid. So basically, it's a core of either DNA or RNA with a protein covering over top of it. The nucleic acid and the capsid are also called the nucleocapsid. Some have a phospholipid envelope, some do not. The outermost layer is what provides protection and recognition sites for host cells. In the intracellular state, meaning once it goes into the actual host cell, the capsid is removed and the virus exists only as a nucleic acid. So virions are complete virus particles that include the nucleic acid, the capsid, and in some cases an envelope, and that's what they're showing you in this particular picture. So this is what it looks like when it's not invading a cell. The genetic material of viruses, they show more variety in their nature of their genomes than do bacterial cells. And this is one of the reasons why viruses are so hard to find treatments for. They're harder to study because there's a lot of variations. An example I can give you is the HIV virus. So that virus, it took almost 10 years to figure out how it was doing what it was doing because it uses a completely different process that scientists had never seen before. So viruses are really tricky. They have a lot of different tools in their tool chest to try to get their way into host cells. The primary way scientists categorize and classify viruses is by their genetic material. So they can be, as I've said, DNA or RNA. They're never both, but they're one or the other. But they can be double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA, which is completely different than what we've looked at with any other type of cell. They can have a linear or segmented chromosome. It could be circular, it could be single. So like I said, there's a lot of variations here. Uh, the genome itself, though, is much smaller than what we see in cells because it's really only carrying copies of how to make more viruses. So it's not carrying copies about how to make proteins and all the other things that most cells have in their genetic material. This one just has replication information, so it's pretty small. What about the hosts? So most viruses affect only a particular host cell. So they don't infect everything. So for instance, when you get a respiratory virus, it only infects your respiratory cells. It doesn't get into your digestive cells, it doesn't get into your urinary cells, anything like that. So they have a specific host type of cell, and they even have a host range typically, meaning that they only infect certain species. The idea of only attaching to particular host cells is due to an affinity of the viral surface proteins for the complementary surface proteins on the host cell's cell surface. They may be so specific that they only infect a particular kind of cell in a particular host. So they may only go after one type of respiratory cell. There are several types in the respiratory system of a particular host. Generalists infect many kinds of cells or many different hosts. There's not a lot of those when we talk about viruses. For the most part, with viruses, they're pretty specific. All types of organisms are susceptible to some viruses. So there are viruses for everything. I always use the pox virus as an example. There is a pox virus for everything. You and I get chicken pox. Monkeys get monkey pox. Chickens get fowl pox. They don't get chicken pox. Plants can get a pox that is specific to them. For instance, tobacco mosaic virus is specific to certain types of plants. So all organisms have viruses that are specific to them. Bacteria have viruses that are specific to them, called bacteriophages. So here's some examples of plant, bacterial, and human hosts of viral infections. So just about everything can get viruses. Viruses are really, really small, or virions in general are very small. We're looking at, in the top box, we're looking at a red blood cell, and you can see the size of that. And next to it, the little teeny tiny, what looks like a little uh, cigar there, is an E. coli bacteria. Teeny, teeny, tiny. And even that, as small as it is really huge compared to what's sitting next to that. And that's the smallpox virus, is the little thing that looks like a little pincushion or a porcupine there, or maybe a little hedgehog. And then a bacteriophage next to it. And then even smaller things like a bacteriophage, MS2, teeny tiny, a poliovirus. So they get teenier and tinier. Viruses in general, we can't see underneath them by light microscope like what we would use in our labs. They're going to require special types of instrumentation. Bacteria, yes, we can see those with proper staining techniques that you guys have all learned. But viruses, that's not how it works for them. Let's talk about the capsid morphology, the shape of the capsid. So this is what provides protection to the nucleic acid that's inside. It has a means of attachment to the host cell. That's the whole idea. You got to attach to the host cell so you can insert your genetic material into that host cell as a virus and let the host become the machinery for making all of the new viruses. It's composed of protein subunits that are called capsomeres. Those capsomeres may be made of single or multiple types of proteins. The shape of the virus can be classified by its virion shape. There are three basic types of shapes that we look at. There's helical, there's polyhedral, and there's complex. So underneath of a scanning electron microscope, viruses are actually kind of pretty. There's nothing nice about a virus, but nonetheless, these are kind of interesting looking. Let's look at a bacteriophage T4. This is a virus that specifically attaches itself to bacteria. And it looks like something that would land on the moon. So it has the head of the virus up at the top portion there, and that's where the genetic material is. And then you have the tail, and notice that you have the tail fibers, and then you have a little spiky base plate, and that's how it attaches itself to the outside of the cell. And then literally it uses that tube in the middle to insert, almost like an injection, insert its genetic material into the host cell. The viral envelope is acquired from the host cell during viral replication or release. The envelope is a portion of the membrane system of the actual host that it's attached itself to. It's composed of phospholipid bilayer and proteins. Some proteins are virally coded glycoproteins. We call those spikes. There's a lot of talk about viral spikes. When we talk about COVID. And so the spikes are part of what attaches to the cell itself, and those can change, and that created a big problem with COVID. Envelope proteins and glycoproteins often play a role in host recognition. The envelope viruses are more fragile than naked viruses. So here it's showing you an enveloped virion. So um, it's got this little capsule around the outside of it, this little envelope, this little structure around the outside of it. All right, a nice table that shows you just how novel viruses are, how different they are from cells that we have been talking about previously. So just for example, comment number 1, for viruses, they are inert macromolecules outside of a cell, but become active inside of a cell. And then we compare that to regular cells, like we've been talking about, bacterial cells, those kinds of things. They are able to metabolize on their own. Another example, viruses do not grow or divide. Cells we know can engage in mitosis, or they can engage in binary fission. We know that they have ways of dividing and growing. Viruses are acellular, whereas cells are cellular, meaning acellular, meaning they don't have all the components of a true cell. So you can look through this whole list of things and see how they're different, and get an appreciation for how very different a virus is from a cell. And that's a really important idea to grasp, because how we treat viruses in the lab and in the clinic are going to be really different than how we treat cells, like a bacterial infection. All right, how do we classify viruses? There's several different things that are looked at to make that classification. The type of nucleic acid, meaning is it single-stranded, double-stranded DNA or RNA, the presence or lack of presence of an envelope, the shape and the size. Viruses have only been organized into families. The relationship among viruses is not well understood by taxonomists. This is an area where there's a lot of work, and I'm sure a lot of changes that will be coming in the upcoming years as they research and learn more about viruses. So here is some examples of human viruses and how we organize them. So the poxviridae is the very first one on there. This is a double-stranded virus, and the representative example here is smallpox or orthopox virus. And so you can take a quick look through here. Some of these you may already be familiar with and have already heard about. Others you'll be learning about. There's some more of them. So there are a lot of families that we see in human viruses. Okay. So I don't require that you learn all of those different families. The ones that I talk specifically about in the lecture, of course, I want you to know about, but don't memorize that whole chart. You can always look that up. Viral replication. So as I mentioned already, for a virus to replicate, it is dependent upon the host and its organelles and enzymes to produce new virions or new particles of viruses. There's a couple of different ways that it's going to happen. We're going to talk about the lytic replication system first. This is viral replication that usually results in the death and lysis of the host cell. There's 5 stages to the lytic replication cycle. There's attachment, entry, synthesis, assembly, and release. Okay, so here we are looking at a bacteriophage, a virus that infects bacteria, and the lytic replication cycle it undergoes. So it's showing you the attachment up there at the top. It uses those tail fibers to attach to the outside of the bacterial cell, and then it literally, almost like a hypodermic needle, as I said before, it inserts its genetic material into the host cell, and then that is going to be incorporated into the host cell's genetic material, and it's going to start to make copies of it, just like it was making copies of whatever it's making copies of before. And then it's going to fill up the inside of that bacterial cell to the point where it literally ruptures, and all of the new bacteriophages escape and go infect another host. So here we are looking at a graph of virion abundance in the lytic cycle. And over on the left-hand side, we're looking at the number of infective virions in the medium, and across the bottom, we're looking at time in minutes. And so you can see from the point of entry into the first 5 minutes, it has already started to synthesize and assemble. And within 25 minutes, it is causing that bacterial cell to rupture. So this process does not take very long at all. And so here it is again. So you've got, the attachment, and then it inserts its genetic material in. That's the green chromosome that you see there. It goes and incorporates that right into the chromosome of the bacterial cell, which is the purple one, and it now becomes nothing short of a virus machine. And so it makes a whole bunch of those bacteriophages, and it ruptures all of those out, and they go attack another cell. There is another version of viral replication called the lysogenic replication of bacteriophages, and this is a modified replication cycle. The infected host cell grows and reproduces normally for many generations before it lyses. This occurs with temperate phages. In this case, we create what's called a prophage that's sitting inside of the cell in the inactive state. Lysogenic conversion is what happens when the phages carry the genes that alter the phenotype of the bacteria. So it infects the host cell and it inserts its genetic material, but the effects on the cell are not immediate. It has now become a virus factory, and so it's making lots and lots of copies because it has altered its DNA or RNA, and over time, it's eventually going to rupture, but until that time, the prodigy that it's making have this new, improved, if you will, version of the genetic material. Just showing you a bacteriophage called lambda. So this is the lysogenic replication cycle in bacteriophages. You can see all of the steps with a little blue square. So there's attachment and entry. Then there's the prophage that's being created. Then there's replication of that chromosome and the virus. When we have lots of cell division, so now the new bacteria that go out have this altered genetic material, and then eventually it goes into the lytic cycle. The cell goes into the lytic cycle, but not until it's made lots of copies of itself and sent it out there. So now let's move along. We've been talking about how viruses infect bacteria. Now let's talk about how viruses infect animals. So we have the same basic replication pathway as the bacteriophages, but there are some differences. There's the presence of the envelope around some viruses. There is the eukaryotic nature of animal cells that we have to recognize, and the lack of a cell wall in animal cells that we have to acknowledge. So we have the attachment of the animal virus. This is a chemical attraction between the viral protein and the cell receptor on the host cell. Animal viruses don't have those fancy tails or tail fibers that we saw with the bacteriophages. They have glycoprotein spikes or other attachment molecules that are going to allow for this attachment. The entry and uncoating of the animal virus is a little bit different. It can enter the host cell by at least 3 different mechanisms. One is direct penetration, another is membrane fusion, or through endocytosis. So showing you those 3 different ways that the animal virus is actually going to get into the cell. Synthesis of the DNA viruses of animals. Each type of animal virus is going to require a different strategy depending on its nucleic acid. DNA viruses often enter into the nucleus. RNA viruses are replicated in the cytoplasm. Things that we have to consider. how messenger RNA is synthesized, and what serves as the template for the nucleic acid replication. Double-stranded DNA viruses, they are similar in their replication of cellular DNA. The viral genome is replicated in the nucleus, and then the viral proteins are made in the cytoplasm, just like we've seen with eukaryotic cells. There are some exceptions. Hepatitis B virus replicates its DNA from an RNA intermediary. Remember I said lots of complicated little extra things that happen with viruses, which is why they are so incredibly hard to study. They don't follow the same pattern. Single-stranded DNA viruses, cells don't use single-stranded DNA. Parvoviruses have single-stranded DNA genomes. The DNA strand folds back on itself to form a double-stranded RNA or DNA, which is replicated by the cellular DNA polymerase. So another little twist in the process. The newly replicated strand is released as a single-strand DNA. How are RNA viruses synthesized with animals? The synthesis of RNA viruses differs from DNA viruses. Positive sense viral RNA can act as messenger RNA, so the template to make the proteins. Negative sense viral RNA cannot be directly translated. There's 4 types of RNA viruses. There's positive single-strand RNA, there's retroviruses, there's negative single-stranded RNA, and there's double-stranded RNA. Viruses are tricky, y'all. I'm telling you, if you decide to be a virologist, you have your work cut out for you because there are so many anomalies. So here it's just showing you some of the different ways that proteins are synthesized with the different types of RNA viruses, animal RNA viruses. So positive sense single-stranded RNA virus is in the very first one, and in the middle one you have a negative sense single-stranded RNA virus, and the last one you have a double-stranded RNA virus. And they all do what they do differently. The end result is the same, but it's just how they have maneuvered a way to get into the cell. What about they don't use their genomes as messenger RNA. They use a DNA intermediary that's transcribed by viral reverse transcriptase, an enzyme, as the template to produce viral genomes. This was the complication in HIV because it uses reverse transcriptase. It uses that particular process. It's a retrovirus, and that's where they first really started to understand about retroviruses. It actually took them almost a decade to figure out how that particular virus was doing what it does. So here are the different synthesis strategies for animal viruses. Again, a lot of information. Viruses are just super tricky. How are the viruses assembled and released? Most of the DNA viruses assemble in the nucleus. Most of the RNA viruses develop solely in the cytoplasm. The number of viruses produced depends on the type of virus and the size and initial health of the host cell. Envelope viruses are going to release via budding, and they can result in persistent infections. Naked viruses are released by exocytosis or lysis. So here's just a picture of the process of budding in an enveloped virus. So you had the virus capsid, the viral capsid in one there. You had the little yellow, the little red spikes out there are the viral proteins. Then you had a budding of that enveloped virus, and then it pinches off and it goes off to go do its dastardly deed somewhere else. A graph looking at the pattern of virion abundance in persistent infections. So persistent infections are ones that you just can't seem to get rid of. And so we're looking at, on the left-hand side, the number of infective virions in the medium, and then time, looking at time across the bottom. So the point of attachment to the entry, to the synthesis, to the release, and then you see how high up that graph goes, representing the number of infected virions over time. That's why it's considered persistent. A lot of times in viruses, they're fast to come on and make you sick, but they're really quick to clear out. A week or two. Now, there's exceptions to that, obviously, hepatitis virus and AIDS and all that sort of thing. We're talking about colds and those kinds of things. They come and go pretty fast. Persistent infections are ones that are going to last a long time, and this is exactly why. Okay, so this is a video. It's a little bit long, but it's a nice review of everything that I've been talking about. So I want you to stop the recording and touch anywhere on the screen, and you should be able to play this video. If not, it is in your textbook as well. So I'm not going to play all the way through this because it's pretty long and it'll make this presentation longer and take forever to upload to YouTube. But you need to stop and listen to this because it'll really summarize everything I've been talking about so far. All right, what about latency of animal viruses? When an animal virus remains dormant in a host cell, the viruses are called latent viruses or proviruses. They may be prolonged for years with no viral activity, so they just sit in there. The incorporation of provirus into the host DNA is permanent. This is not something that's going to go away. So here's a nice table that shows you the comparison of bacteriophages and animal virus replication. And so we talk about the attachment, penetration, uncoating, cytosynthesis, site of assembly, mechanism of release, and nature of chronic infection. The role of cancer and viruses. So cell division is under a very strict genetic control. So when we talk about mitosis and we talk about PMAT and we talk about the idea of how the cell goes through that whole process, we also recognize that a cell only divides when a cell has instructions to divide and only when it needs new cells. So genetically, this is all genetically engineered. Genes dictate that some cells can no longer divide at all, and cells that can divide are prevented from unlimited division. And that's what I mean by it dictates when this is going to happen. Genes for cell division turn off, or genes inhibiting cell division can be turned on. So that's important to understand. This is all genetically controlled. A neoplasm is an uncontrolled cell division in a multicellular animal cell. So when cells divide out of control, a mass of neoplastic cells is called a tumor. So what a tumor is the result of uncontrolled mitosis. When we talk about benign versus malignant tumors also are called cancers. Metastasis occurs when those tumors or cells from those tumors break off and spread to other areas of the body. Proto-oncogenes promote cell growth and division. Uncontrolled activation of oncogenes can lead to cancer. So this is uncontrolled mitosis. Environmental factors that contribute to the activation of oncogenes include UV light, radiation, carcinogens, and viruses. So there's something called the oncogene theory of the induction of cancer in humans, and it's showing you what in the normal state, how things are supposed to happen. We have repressor genes, we have oncogenes, and they're supposed to keep things in check. The first hit is when we have a virus that inserts a promoter into the oncogene and gets that oncogene geared up to go. The second hit is when the virus inserts a barrier into the repressor gene and keeps it from stopping the process. Viruses may cause 20 to 25% of human cancers. They carry copies of the oncogenes as part of their genomes. They promote oncogenes that are already present in the host. They interfere with tumor repression. Specific viruses are known to cause some human cancer, including Burkitt's lymphoma, Hodgkin's lymphoma, Kaposi sarcoma, and cervical cancer. Viruses cannot grow in a standard microbiological media. So all of the stuff that you've been learning in lab about how to grow bacteria does not apply to viruses. We cannot do those in the lab. They don't work the same way. They must be cultured inside of a host cell because they require a host cell for replication. There's three types of media for culturing viruses. This includes media that contains mature organisms, embryonic eggs, and cell cultures. So how do we culture viruses in mature organisms? We do this in bacteria. So the phages grow in the bacteria in liquid culture or on the agar plates. The lysis of the bacteria is going to produce a plaque. This allows for the estimation of the phage numbers by plaque assay. This is not something we ever do in teaching labs, by the way. So this is what the viral plaques on a bacterial lawn looks like on the surface of an agar plate. So the places that are clear are the plaques. All of the yellowishy, goldy stuff, that's all the bacterial lawn or the growth of bacteria in that particular area. And again, these are bacteriophages, so they're attacking the bacteria. What about doing this for plants and animals? There's numerous plants and animals that have been used to culture viruses. The laboratory animals can be difficult and it is expensive to maintain. And of course, there are lots of ethical concerns associated with doing this with animals. How about using embryonic chicken eggs? This is an inexpensive method and it is because an egg is one of the largest cells that we have. So a single egg is actually a cell, right? It's free of contaminating microbes. It contains a nourishing yolk. Fertilized chicken eggs are often used as embryonic tissues to provide the ideal site for growing viruses, and some vaccines are prepared in chicken cultures. So if you go get a vaccine, they always ask you, are you allergic to eggs or chicken or anything like that? And this is why, because they've grown that virus in a chicken egg. So just showing you how they do this, and they use an embryonic chicken egg. So this is a fertilized chicken egg that they're doing this in. A little bit about culturing viruses in cell culture. The cells are isolated from an organism and are grown in a medium or in a broth. The cell cultures sometimes are inaccurately called tissue cultures. They're not. They're just cells. They're not tissues yet. There's two types of cell cultures, the diploid cell cultures and the continuous cell cultures. So again, high-end stuff that we do not do in teaching labs. This would be something you would be doing in a research lab, a pharmaceutical lab, something like that. You wouldn't be doing this in an everyday lab, but they're showing you how they do cell culture research. So back to what I said in the very beginning, are viruses alive? Because I mentioned that they live in this kind of quasi no man's land. They're not really technically alive. Some consider them to be complex pathogenic chemicals. Chemical is not a cell. Others consider them to be the least complex living entity. They use sophisticated methods to invade hosts. They have the ability to take control of their host cell. They're able to replicate themselves once that happens, and they do evolve. So that's why some people say they are the least complex living entity. They don't still stop short of calling it a cell. Other people refer to them as an obligate intracellular parasite, meaning that they have to get inside of the host cell. So there's a lot of ways people refer to it. Now let's talk about some other parasitic particles, the virioids and the prions. So viriards are extremely small circular pieces of single-stranded RNA genetic material that are infectious and pathogenic in plants. These are similar to RNA viruses, but they lack a capsid. The virioid RNA does not code for proteins. The virioid RNA adheres to the complementary plant RNA. The plant enzyme is going to degrade the double strand of RNA and result in a disease state. So we don't really study plants in here, but I do want you to be aware that virioids exist and they are what attacks plants. Here's an effect of virioids on plants. These are potatoes. and I'm not sure which potato disease they have, probably potato blight. But nonetheless, you can see what happens to the growth of the potatoes and the morphology of the potatoes because of the virus. Okay, let's talk a little bit about prions. These are the other parasitic particle. Proteinaceous infectious agents. There's two forms of this. There's the cellular PRP, which is made by all mammals. This is a normal functioning structure that has alpha helixes. So you make these in your brain all the time. There are also prion PRPs, which are disease-causing forms of a cellular PRP that has a misfolding. It's folded as a sheet. And so prion PRPs cause cellular PRPs to refold and become this horrible thing called a prion PRP. So a drawing depicting how they look differently, a cellular PRP compared to a misfolded prion PRP. And so just giving you the step-by-step process of how it happens. There's just a misfolding that occurs. But the effects are devastating. So this all happens in the brain, and what happens is you end up with a brain that has brain tissue that has holes in it. And so they call that spongiform encephalopathy when that happens. So normal brain tissue on the left-hand side, and then brain tissue that has prions in it. So prion diseases. So this can just happen because sometimes it just happens. But there are also ways that you can contract prions. Okay. And one of those is called spongiform encephalopathies. These are large vacuoles that form in the brain, has a characteristic spongy appearance like we saw in the previous slide. So BSE, scrapie, kuru, CWD, and variant CJD are all diseases that are in this category, and they're super important for you guys to understand because they often deal with livestock. So they're transmitted by ingestion, transplantation, or contact of mucous membranes with infected tissue. There's no standard treatment for any prion disease. So an individual has to ingest infected tissue from an infected animal. And that individual could be another animal, but it also could be a human. What makes these guys so scary is that normal sterilization procedures do not deactivate prions. Prions are destroyed by incineration or autoclaving in concentrated sodium hydroxide, or lye. And the European Union recently approved the use of enzymes to remove prions from medical equipment. So this is where this is of concern. When you're doing any type of surgery and you're using equipment in your lab to do that, or in your operating room to do that, you can pass this on to the next patient if the instruments are infected. I want to stop here and say that this is extremely rare. Extremely, extremely rare. So this is just something you need to be aware of, but it's not something that you need to feel like it's going to happen with every case that you do. It's not. All right, so here is a little chart looking at the human prion diseases versus animal prion diseases. Human prion diseases, Jacob's Creutzfeldt-Jakob disease, GSS, fatal familial insomnia, Kuru, and then animal prion diseases, which is more our focus in here. So scrapie, transmissible mink encephalopathy, chronic wasting disease that you see in deer, and bovine spongiform encephalopathy, or mad cow disease. This course is meant primarily for students who are doing vet med, but I do have other students in here from time to time. So here is an extra credit opportunity, and I will post more about this after this week. But If you would like to earn 10 points, up to 10 points on your lowest test score, if you will choose one of the animal prion diseases, if you're a vet tech student. If you're one of my other non-vet tech students, then it's perfectly fine if you choose a human prion disease. Or if you're just really, really wanting to know about a human prion disease, even though you're a vet tech, you can go ahead and choose one of those two. But I would prefer you focus on animal prions. And you need to write me a short essay. I'm going to put more information out there about it. I don't want you to copy and paste it from somewhere. I want you to actually go out and read about it and find out. And I want you to write me an essay as if you are explaining it to a patient or to the caretaker of an animal that has one of these diseases. So I'll post more details about this on our uh, Brightspace page. But I want you to start thinking about which one you think you might want to be interested in. So finally, we have some charts here that are comparing viruses, viriyoids, prions to bacterial cells. There are some very significant differences. We lump them all together kind of in this course, but I want you to see that they are very, very different from each other, and the outcomes for your patients and the treatment for your patients are going to be different. All right, that is it for this particular chapter. Hope it didn't scare you too much with prions, because they are kind of a scary thought. Make sure you have done your homework in Pearson and taken the quiz for this particular chapter, and I will see you for the next one.